Hi, this is part 3, and I'm going to talk about RNA processing. So before RNA can actually exit the nucleus and go to the cytoplasm where it can be translated, RNA has to be processed. So there are several steps to RNA processing. Like I said, in eukaryotes, in human cells and plant cells and animal cells, mRNA must first exit the nucleus and then go to the cytoplasm. This is because transcription takes place in the nucleus and translation takes place in the cytoplasm. Recall that nucleus is an envelope and our genome is being stored in the nucleus, and so all mRNAs get transcribed in the nucleus, transcription occurs in the nucleus, and then mRNA exits and goes to the cytoplasm. So there are several steps that take place during mRNA processing. So initially when mRNA is synthesized, it's called pre-mRNA, and once it gets processed, we call it mature mRNA. mRNA can also be called a transcript. So sometimes we call it a nascent transcript when it's a pre-mRNA. And once it gets processed, we call it a mature mRNA transcript or mature transcript. So what are the steps in processing? And before we go on, I just wanted to say this is kind of similar when you think about it as an analogy to a company. So suppose you have some kind of product that you want to sell on Amazon or Alibaba Express or somewhere. So before sending it to the customer, you have to process it by putting it in a nice box and adding marketing materials and so on and so forth. So you don't want to just send it out in its raw state. You want to make it look pretty before you give it to the customer. So it's kind of an interesting analogy, I think, and here it applies to this also, because here mRNA has to be processed before it can go to the cytoplasm. Obviously, it's not to make it look pretty. I will explain the reasons why it has to be processed. So first, the methylated cap is added to the 5' end of the mRNA transcript. Specifically, this is important so that mRNA can be recognized by the ribosomes and it can be translated. So it's a recognition site for protein synthesis. So at the 5' end of mRNA, there's a G nucleotide, a guanine nucleotide that gets added, and it has about 7 methyls on it. So it's a methylated G nucleotide that gets added to the 5' end. A methyl is a CH3, so you have 7 CH3s added to the G nucleotide on the 5' end. Okay. And on the 3' end, a poly A tail is added. So it's just a stretch of A's, and it could be anywhere from 50 to 250 A's, adenine nucleotides added to the 3' end. So this is also necessary for protein synthesis. This stabilizes mRNA and prevents its degradation by the enzymes in the cytoplasm. Many of you who are taking this course are seniors, so if you wanted to think about it as cap and gown analogy, so mRNA must get its cap, which is a methylated G nucleotide, and it also has to get its tail. Okay. The next step in RNA processing is a process called splicing. Splicing occurs when introns or intervening sequences are removed and exons are kept. Exons are part of mRNA that remain and which are later translated into amino acid sequences. So it turns out that introns are removed and exons are spliced together by joining the ends of the exons. Note that introns sometimes may outnumber the number of exons, so you might have more introns than exons, and introns might actually be longer sequences. They might have more base pairs in them than exons, meaning that they might have more bases, more ACTs and Gs in them, so they might be longer sequences, but yet they get removed because they are not coding sequences. So once the process of splicing is complete, the mature mRNA is sent out to the nuclei. And just to go to this picture right here, so here we have exons and introns, so exons and introns, and you can see that in the pre-mRNA you have both, but after splicing you just have the exons. And I'm gonna show you a short video now which describes how splicing is done in the cell. Essentially you have boundaries between exons and introns with specific sequences which are recognized by the spliceosome. The spliceosome is composed of RNAs, small RNAs and proteins, and those are known as not Smurfs, those are known as snRPs. snRPs, and also known as snRPs. And these are, again, short RNA and small protein complexes. And they bind at the intron-exon junction, they recognize those specific sequences, and then they make sure that the intron forms a lasso structure and gets excised, and exons are kept.
Most eukaryotic genes are composed of numerous short coding sequences called exons, interspersed between long stretches of non-coding sequences called introns. The initial messenger RNA molecule copied from a gene includes introns as well as exons. Before this RNA molecule can be used to make a protein, the introns must be removed by a process called Rna splicing. A structure called a spliceosome plays a key role in RNA splicing. The spliceosome is composed of several small molecules called snRNPs, or Snurps. Snurps are made up of protein and small RNA molecules. Formation of the spliceosome begins when one kind of Snurp binds to the 5' end of an intron, and a different kind binds to the 3' end of an intron. Additional Snurps then interact with the complex, bringing the two ends of the intron together and causing the intron to form a loop. This also gathers all of the Snurps together as the completed spliceosome. The intron is then cut out and the exons are joined or spliced together. The spliceosome then falls apart as the Snurps are released.
Now, it turns out that there's something called alternative splicing. Alternative splicing is a mechanism by which different exons can get spliced together. So alternative splicing is a mechanism of combining exons of a gene in different ways. Cell types can use versions of the same protein in slightly different ways in different tissues. So before, remember we talked about the dogma of molecular biology, and we said that DNA makes RNA makes protein, and we said it doesn't always hold. For example, in retroviruses, RNA can be transcribed back to cDNA using an enzyme called reverse transcriptase. So this was a challenge to our dogma. Also, we said that genes are protein coding regions of the genome, but now from this example on splicing, we know that a gene also contains non-coding sequences called introns. So now we define the gene as a sequence that contains information needed to make protein. So it's not just the coding sequences, but the introns can serve as regulatory sequences during transcription. They could serve as enhancer elements or as binding sites for repressor proteins. So basically, introns are important for transcriptional regulation. So the definition of a gene has changed in that way. So it's not just the coding region of the genome, but it contains both exons and introns. Also, from the model by Biddle and Tatum, who, remember from our previous lectures, I said that Biddle and Tatum were a team of they were basically two scientists who worked on Neurospora fungus, and they realized that a single gene codes for a specific single protein. So a gene codes for a specific enzyme important in a certain biochemical pathway. But now we know that one gene can actually code for multiple versions of the same protein because of alternative splicing. And this is why we have only 30,000, well, about 24,000 genes that are known, and perhaps we have more genes that haven't been discovered yet. So suppose we have about 30,000 genes, but we have 200,000 proteins, and this is because of alternative splicing and alternative transcripts being produced from one gene. So for example, one gene could produce about on average, 5 different transcripts, alternative transcripts. So here's an example of this. You have, exon 1, 2, 3, and 4, and 5, and you can get several alternative transcripts. Like in one on the left, you have 1, 2, 3, 4, and 5 combined, and in the other one, it's 1, 2, 4, and 5 combined. And in the one on the right, you have 1, 2, 3, and 5 exons being combined. So you have alternative transcripts which will give rise to alternative proteins, which might be tissue specific. Like in the liver, you might have protein A being expressed, which comes from transcript 1, and in the brain you might have protein B being expressed, which comes from transcript, the second transcript, which was created by joining exons 1, 2, 4, and 5 together. So here's another example. So there's this protein called calcitonin, which is important in how calcium is being used in the body. And it turns out that calcitonin gene can produce two alternative transcripts. One of them is expressed in the thyroid, and another one is expressed in neuronal cells in the brain. And so you end up with two different transcripts which give rise to two different proteins. Interestingly, in patients with Williams syndrome, and if you recall, we talked about Williams syndrome in the very first class. So these are patients who have some cognitive deficits. They have difficulty with spatial reasoning. They also have specific facial morphology. So, and it turns out Williams syndrome patients, children with Williams syndrome, are born with very high levels of calcium. They have hypercalcemia, which might contribute to the kind of difference we see with their facial dysmorphology and bone structure differences and dental issues that they have. So basically they have this hypercalcemia. It also affects their cardiovascular function. So basically, so why do they have such high levels of calcium? And the idea might be that they might have a mutation at the splicing site, which results in how calcitonin protein is made, and that can really affect calcium levels in the body. So why is calcitonin interesting? Because it affects many different functions. For example, High levels of calcitonin can cause migraines, so there's currently a few drugs on the market which are antagonists of this calcitonin pathway, and they help treat migraines. Also, you can see calcitonin being important in a number of other um, medical issues like wound healing and hypertension and obesity. And so just to relate this to business, a lot of companies are interested in developing drugs using calcitonin as a target for their drug development process.